Okay, welcome to a new module. In this module, we're going to be discussing chemical messengers of the immune system called cytokines, as well as some of the receptors that allow the immune system to detect those non-self entities that we've been discussing, in particular pattern recognition receptors. But we're going to start talking about cytokines, and in this lecture, we're going to introduce ourselves to general principles of how cytokines signal, how they transmit immunological messages between cells within the immune system and in cells in the rest of the body. So, cytokine signaling in general looks pretty similar to most other types of chemical messengers that you may have studied in other classes. You can kind of think of cytokines as being analogous to neurotransmitters of the nervous system or to hormones of the endocrine system. They're secreted soluble molecules that activate receptors on the surfaces of cells and then transduce some kind of signaling depending on what type of receptor it is. So, in general, there are actually many different kinds of cells that can secrete cytokines, not only immune cells, although immune cells are very good at secreting cytokines. But cytokine secretion is going to happen downstream of some kind of inducing stimulus. This inducing stimulus might be infection, it might be a different cytokine, it might be injury, but something is going to turn on cytokine expression in some sort of cell in the body. And when it does so, it induces transcription of cytokine encoding genes, and ultimately they're translated and secreted. And these secreted cytokines then are going to bind to their cognate receptors on the surface of downstream cells. And activation of these cytokine receptors is in turn going to induce either new gene expression or activation of enzymes, for example. And the things that are activated in the second cell are usually the proteins that are going to perform the biological effect or the effector function of that cytokine. So it might be something like inducing proliferation in the case of clonal expansion of T cells, differentiation, maybe like a plasma cell differentiating from a B cell death in the case of cytotoxic T cells. They use cytokines and other molecules like cytokines to induce cell death. These are just examples of the many, many different things that happen downstream of cytokine receptor activation. And so here we sort of see cytokine signaling happening between two cells, but cytokine signaling can really happen all over the body. So it can happen in endocrine fashion. So cytokines secreted by the original cell can enter the circulation where they signal to distant cells in other parts of the body. So let's say, for example, you had an infection in your hand. Cytokines from immune cells and infected cells within your hand are going to signal all the way back to your lymph nodes and your spleen in order to activate immune responses. And so that long range activation or cytokine signaling is happening in what we call an endocrine fashion. However, cytokines also signal locally, very importantly. And so, on a local scale, a cytokine signaling between two or more cells is called paracrine. So, when one cell is activated, secretes cytokines that are sensed by a different cell in its environment. That's a paracrine signaling. In contrast, cytokines can also signal in an autocrine fashion. So when a cell becomes activated and secretes cytokines, those cytokines can then bind to receptors on the surface of the same cell. And many cytokines also signal in an autocrine fashion. So all three of these have their roles within immune responses, but these are sort of three general principles for the different scales at which cytokine signaling can be transmitted in the body. There's some additional concepts that sort of organize the way that cytokines interact with the different cells that they target. A major idea in cytokine signaling is this concept of pleiotropy. So pleiotropic cytokines, and there are many, are capable of inducing a lot of different effects in a lot of different cell types. So they just have really, really diverse outcomes. They can do all sorts of things. So your book gives an example. Actually, this is a different textbook, but your book probably also talks about it. But an example here in this figure, nonetheless, are activated Th cells, so T helper cells. These are CD4 T cells, and they express a cytokine called IL-4. IL-4 is a highly pleiotropic cytokine because lots of different cells express the IL-4 receptor. So IL-4 can signal onto lots of different cells, and when it does so, it can induce lots of different effects, anything from activation to proliferation to differentiation. All of these are distinct biological outcomes. And so this example of IL-4 is just one. There are many pleiotropic cytokines, but the big idea is that pleiotropic cytokines have all sorts of diverse outcomes outcomes in lots of different target cells in which they can induce their signaling. So, in addition to pleiotropy, cytokines also exhibit significant redundancy, meaning that some cytokines can do the same thing. So, IL-2, IL-4, and IL-5 are related cytokines, and all of them can induce proliferation, for example, in B cells. And so, there are lots of reasons why we've evolved significant redundancy in our cytokine signaling networks. One is that sometimes we have random mutations during development or during gestation or whatever, and we lose one or more cytokines. And so, it's common to have SNPs or small mutations that make us lose function in a random gene. And so, for things that are really, really important for our survival, like the proliferation of B cells, we really want to make sure we have multiple redundant sort of fail-safe mechanisms to induce those processes. And so, IL-2, IL-4, and IL-5 are all sufficient to induce the same process. So if we lose one, we have some backup. Mutation is not the only example of where we might lose one of our cytokines. Many pathogens, for example, encode endogenous inhibitors of cytokines. So if a virus encoded an inhibitor of IL-4, for example, we would have backup cytokines to be able to do some of the things that IL-4 does. That's another example of why redundancy is often important. Cytokines also induce something we call synergy, and that name should be somewhat self-explanatory. But in this case, these are effects that happen, they either require two or more cytokines to be signaling at the same time, or they're at least enhanced by the presence of two or more cytokines. And so an example of this process is a process that we'll discuss in a later lecture called class switch recombination of B cells. So this is a process by which B cells determine which type of antibody they're going to secrete. We'll talk about that a lot in the future. But for our purposes here, what I want to show is that it really requires both IL-4 and IL-5 signaling at the same time to get a really strong class switch recombination effect. And so IL-5 synergizes with IL-4 to induce this type of biological activity. And these kinds of things are important for immune processes which we want to keep under careful regulation. We don't want them sort of happening accidentally or all the time. And so you can almost think of it like, two-factor authentication or something. You need both IL-4 and IL-5 to induce this process. And again, this is just one example of a synergistic outcome of cytokine signaling. Okay, so synergy is one thing. The kind of opposite of that is cytokine antagonism. So sometimes cytokines synergize, sometimes they actually antagonize each other. So this process of class switch recombination downstream of IL-4 is actually blocked by a different cytokine called interferon gamma. So depending on which cytokines are being expressed at the same time, sometimes they might work together and sometimes they might actually inhibit each other. And this is really important, again, for making sure that whatever immune responses we're generating are carefully regulated and carefully adapted to exactly the parameters of the infection that we're seeing and sort of where in the timeline of infection we are, because these cytokine signals will change in a temporal fashion following the infection. So, pleiotropy, redundancy, synergy, and antagonism, four general principles of the mechanisms by which cytokines influence the immune system and immune responses. So we can talk about a couple or more than a couple. There are actually 6 cytokine families that all of the cytokines in our body sort of fall into. These families are mostly unified by sort of structural and functional features, although as we'll see, these families are a little bit loose and all these cytokines sort of cross-signal with each other. But it at least helps us sort of identify major structural and functional themes that tie together some of the cytokines that we'll discuss in the course. So the first family is the interleukin-1 family. So you'll notice that many cytokines are named interleukins or IL. This name comes from the idea that we're signaling between immune cells. So inter meaning between, leukin referencing white blood cells, leukocytes. So interleukins are signals that signal between white blood cells. And so the first family is the interleukin-1 family. Its sort of cardinal members are IL-1 alpha and IL-1 beta, although there are others. And a big idea about the IL-1 family is that they are very strongly pro-inflammatory cytokines. So they induce really strong inflammatory responses. And we'll talk about each of the families of cytokines kind of on their own terms in subsequent lectures. Right now, we're really just introducing the families, but that's one big takeaway from the IL-1 family. The next big family that we'll discuss are the hematopoietin or class 1 cytokine family. And there are a lot of class 1 cytokines. There's not really a lot to say that ties them together other than they sort of have similar receptor signaling features. And so we'll talk about hematopoietin or class 1 cytokine receptors. But generally, lots of cytokines fall in this family, and they do all sorts of pleiotropic things. So there's not really a lot else to say to sort of explain what they are. We'll just discover them as we talk about them one by one. Okay, the next family are the class 2 cytokines, more commonly referred to as the interferons. Interferons are represented by interferon alpha, beta, and gamma, as well as some additional molecules which have the IL designations. The really important thing about interferons is they are really potent antiviral cytokines. So interferons get their name from their ability to interfere with viral infection. And these are cytokines which come up over and over again because they not only really promote antiviral responses, but they also modulate immune responses in really profound ways. So we'll spend some time talking about interferons as we go through each of the families one by one. Our next family is the tumor necrosis factor family, or TNF. You can notice that TNF alpha and TNF beta are TNF family cytokines, as well as a molecule that's important for CD8 T cells called FAS. But I think a couple things that we'll focus on for the tumor necrosis factor family is that they're somewhat unique in that they can signal either through soluble or membrane-bound forms, and they're also really important cytokines for regulating the processes of programmed cell death. So we'll talk a little bit about apoptosis signaling downstream of TNF when we talk about this family. Another family is the IL-17 family, the interleukin-17 family. These are kind of some of the most recently discovered cytokines. We're still really figuring out exactly what IL-17 is for, but one big idea is that it's really pro-inflammatory again, and actually IL-17 has been associated with a lot of different inflammatory diseases, and so it's a really interesting drug target under development right now for a lot of different diseases. But because it's sort of recently discovered, there's not as much to say about it. But you can at least know that it exists and that it's a pro-inflammatory family of cytokines. Then finally, one of my favorites, chemokines. These are a family of cytokines that induce chemotaxis and chemoattraction, the movement of cells. So chemokines get their name because they are chemotactic cytokines. Sort of smush that together into chemokines. These are just a few of many, many examples of chemokines. But chemokines are important for moving immune cells around the body and attracting them to sites of infection or to the places and tissues where they need to go to develop. And so chemokines are really, really important for a broad range of immune functions. And so we'll dedicate some special attention to chemokines also. Okay, so these are our 6 families. Now, I want to stress that you don't need to memorize all of the members of all of the families. Some of the families, like interleukin-1, you should be able to recognize that IL-1 alpha and IL-1 beta are IL-1 family cytokines, but you don't need to memorize all the ILs and which family they belong to. We'll talk about major examples of each cytokine family, and those will be the ones that we'll focus on for the purposes of this class. Okay, so we've introduced the 6 families of cytokines. What are some of the things that cytokines do? We'll just talk about a couple of examples in this lecture, again, as general principles of cytokine signaling. A major thing that cytokines do are determine both differentiation and the effector function of immune cells. And so T helper cells are a really great example of that. So we'll focus on them on this slide. So remember that T helper cells, their effector function actually is the secretion of cytokines. They influence other immune cells through signaling through cytokines. And so when we have a naive T helper cell, this is a naive CD4 T cell which hasn't seen its antigen yet. It's in a homeostatic state. It's not doing much, but when it sees its antigen, it can actually differentiate into multiple subsets of T helper cells which are listed here: Th1, Th2, Th17, and Treg, which stands for T regulatory cells. The type of cell that it's going to become is going to be influenced by the cytokines which are in its microenvironment when it sees its antigen presented to it by an antigen-presenting cell. So if that antigen-presenting cell is expressing interferon gamma and IL-12, that's going to induce the naive T cell to become a Th1 T helper cell. If the antigen-presenting cell is expressing IL-4, that's going to induce it to become a Th2 cell, and so on. And depending on which type of T helper cell it becomes, that's going to influence the cytokines that that cell excretes. So T helper 1 cells, Th1 cells, express interferon gamma and TNF, really pro-inflammatory cytokines. In contrast, T regulatory cells, Tregs, express TGF beta and IL-10, which are anti-inflammatory cytokines. T regulatory cells are really important for turning down immune responses when they're sort of getting out of control. So these different T helper cell subsets have different functions in the immune response. And so which one we get is determined by cytokines, and then what those cells actually do is also determined by the cytokines that they secrete. So again, memorizing all the cytokines and which ones they go to is not the point of this lecture. We're highlighting a general point that cytokines influence both leukocyte differentiation as well as their effector functions. Another example of this is with macrophages. Macrophages need to do a lot of different things. They're really important innate immune cells, but they also are involved in adaptive immunity through the process of antigen presentation. So they secrete a lot of different cytokines, and these different cytokines each have different effects, which accomplish some different effector function that macrophages are responsible for. So I'll let you read these different things that the different cytokines do. Again, not necessarily the point here, but one thing that we can note is that these cytokines signal both locally, so autocrine and paracrine, but also systemically in an endocrine fashion. And the systemic effects of cytokines are really interesting because they induce things like fever, some of the systemic constitutional symptoms of inflammatory diseases. So you can make special note of those. But again, the idea that I want to stress is that each of these cytokines has its own part to play in the overall shaping of an inflammatory response, both at the local tissue level and at the body-wide sort of systemic level. Okay, so let's zoom in a little bit on sort of molecular mechanisms of cytokine signaling. I want to highlight that there are really 4 basic steps to cytokine signaling within a cell. We start with the activation of a cytokine receptor. So all of those cytokines that we saw in the table were the different families. They all have their different receptors, which all signal slightly differently. But for the most part, cytokine receptors themselves need a receptor-associated kinase to transmit a signal. So most cytokine receptors don't have their own intrinsic kinase activity or other type of enzymatic activity. Most of them need help from some kinase that they recruit from the cytoplasm. So these receptor-associated kinases, in turn, recruit and activate transcription factors. And transcription factors, of course, do what transcription factors do, which is go to the nucleus and induce the expression of genes. And in the case of cytokine receptors, these are going to be immunological genes, which have some sort of immunological effector function. So this could be anything from inducing proliferation, differentiation, antiviral activity, what have you. But for these genes to be turned on, it's going to require the activation of a cytokine receptor signaling through a kinase, which turns on a transcription factor, which induces their expression in the nucleus. So we can see a really common example of this, particularly in the class 1 and class 2 cytokine families, so the hematopoietin and interferon families. They have a really stereotyped sort of pattern of activation, which involves their cytokines activating a dimerized receptor, so a receptor with 2 receptor chains. When the cytokine binds to the receptor, it induces its dimerization, which brings into proximity 2 receptor-associated kinases of the JAK family. These are Janus kinases, or JAK. The JAK kinases, when they're brought close to each other because of the dimerization of the receptor, that allows them to activate each other and phosphorylate the tails of the receptor chains. These tails of the receptor chains, when they're phosphorylated, then are able to recruit transcription factors from the cytoplasm, in particular transcription factors called STAT, S-T-A-T, which are really important immunological transcription factors. So JAK activation recruits STAT to the complex. Once STAT is recruited, it can then be phosphorylated by the JAK kinases, and when the STAT transcription factor is phosphorylated, it becomes active and dimerized. It can now translocate to the nucleus to drive immunological gene expression. So this process that we call JAK-STAT signaling happens with many, many cytokines. We'll see it over and over again. But it represents a really common sort of theme or pattern of cytokine signaling that really unifies the way that these immunological signals happen in most cells of the body. So this is the way that we turn on a cytokine response and induce an inflammatory gene expression profile. How do we turn it back off? Well, another major thing that I want to highlight is that cytokine signals are often self-regulating, and they do so through negative feedback loops. A really famous example of this, and so this is what we just discussed. We have a receptor activated by a cytokine. It turns on JAK, which recruits STAT and phosphorylates it, which induces immunological gene expression. Well, it's going to induce genes which we've discussed, like inflammatory genes, but it's also going to induce negative regulatory genes like SOX, or suppressor of cytokine signaling, SOX. So when SOX is created and is translated, it actually goes back and is an inhibitor of JAK signaling. So you can see this negative feedback loop. When we make SOX, it inhibits JAK, which basically short circuits the signaling, and now the receptor, the cytokine receptor signaling, is basically turned off. And so the expression of negative regulatory factors like SOX is really important to make sure that we don't create an immune response that's too strong or too activated, because inflammation is damaging to our tissues, and sometimes we need that because we need to damage pathogens. But we can also generate a lot of immunopathology if inflammatory responses are too strong. And so negative regulatory mechanisms like SOX expression are a major way that cytokine signals self-regulate to avoid immunopathology and autoimmunity. Okay, let's summarize these general principles. Cytokines transmit immunological signals. Again, they are the neurotransmitters of the immune system, if you will. They're directly analogous, and they signal both locally and systemically. Both within the tissue and also throughout the whole body through the circulation, for example, or through the lymph. And cytokines have diverse and overlapping functions. They do all sorts of stuff. So we're going to talk about examples of these, but remember that a cytokine that can induce a lot of different effects on a lot of different cell types are said to be pleiotropic. Sometimes when cytokines have overlapping functions and we need more than one cytokine to induce a response, that's an example of synergy. Lots of different terms that sort of organize the functional categorizations of the way cytokines signal. We introduced ourselves to 6 major cytokine families, and they sort of share functional and structural similarities, which we'll get into in later lectures. I want to stress, though, that there is significant crosstalk between all of these families. The families are a little bit loose because they all interact with each other and signal between each other. The point of talking about the families is really just to help us kind of categorize and break down cytokines into major classes, which just helps us sort of digest how many of them that there are. But despite which family we're talking about, cytokines work together to shape the development, the differentiation, and the function of all of the different immune cells that we've discussed in the course. So the microenvironment where a naive T cell or other unactivated leukocyte finds itself when it becomes activated is going to determine if it differentiates and which type of cell that it differentiates into. And the cytokines that activated cell expresses is going to influence its effector function in all of those different ways that we discussed. Finally, receptor signaling by cytokines also fits sort of a stereotypical pattern. Typically, signal transduction happens through a receptor-associated kinase, which activates one or more transcription factors, which actually induce the effector proteins that perform the immunological work that the cell is trying to accomplish. And these responses can be self-regulated through induced suppression, and we discussed suppressor of cytokine signaling, or SOX, as a major example of that. Okay, so this is general principles of cytokine signaling. In the next lecture, we're going to dive a little bit deeper into 2 of the families that we introduced in this lecture, the class 1 hematopoietin cytokines, as well as the class 2 interferon cytokines. See you then.